The evolution of complex eukaryotic cells from much simpler prokaryotic cells was one of the most important events in the history of life. Eukaryotic cells contain several kinds of complex cytoplasmic organelles, including lysosomes, endoplasmic reticulum, cilia, and flagella. Two other organelles, chloroplasts and mitochondria, are even more complicated. For more than a century, cell biologists have wondered how eukaryotic cells containing such complex organelles might have evolved. The fossil record provides few clues to the history of life at the cellular level. The oldest fossils of eukaryotic cells have been found in rocks 2.1 billion years old. Unfortunately, these microscopic fossils don't usually show details of internal cell structure, so they provide few clues as to how today's cells evolved. Studies of living cells, however, have provided several different kinds of clues about cellular evolution. One strong hypothesis developed in the 1960s by Lynn Margulis of the University of Massachusetts proposed that organelles in eukaryotic cells were formed when different types of cells joined in a kind of merger. Margulis called her idea endosymbiosis, which literally means living together within. In recent years, molecular biology has been able to test this hypothesis with new techniques and greater precision. A lot of evidence has accumulated to support the hypothesis that the evolution of important complex features of eukaryotic cells occurred through endosymbiosis. The fact that some cells today contain bacteria and algae living as endosymbionts supports this hypothesis. Mitochondria, for example, are similar in size to bacteria, have their own DNA genomes, synthesize some of their own proteins, and are formed when pre-existing mitochondria divide. Most biologists now agree with Margulis's hypothesis that mitochondria are the descendants of free-living bacteria that took up residence in the earliest eukaryotes. Similar evidence supports an endosymbiotic origin for chloroplasts. The membrane systems of chloroplasts closely resemble those of photosynthetic prokaryotes. This suggests that chloroplasts evolved from free-living photosynthetic cells that paired with the earliest eukaryotes. Let's look at the image and think about what the image shows. An ancient anaerobic prokaryote became eukaryotic when a nucleus formed around the DNA. This cell also gained mitochondria when ancient free-living bacteria took up residence inside the earliest eukaryotes. Some of these cells also developed a symbiotic relationship with ancient photosynthetic bacteria. This lineage became plants and other photosynthetic eukaryotes. Those cells that did not gain chloroplasts gave rise to the lineages of animals, fungi, and other non-photosynthetic eukaryotes. Is it likely that non-photosynthetic prokaryotes could have evolved into chloroplasts? No. Chloroplasts have the ability to carry out photosynthesis,